Hi, my name is Gomez. I'm in the Snyder Lab at Stanford University School of Medicine and Cal State East Bay, Department of Biological Sciences. And I wanted to go over the clinical and gut microbiome composition between responders and non-responders of immune checkpoint inhibitors, which is a cancer immunotherapy. So here we have a study, a cohort CA209-538, where they did, where Gunjur et al. In 2024 did a microbiome analysis where they took a microbiome evaluatable patients, 106 patients of the microbiome that's evaluable. So they evaluated microbiome patients, 106 patients. And that what they looked at was a combination ICI and a nivolumab maintenance. And we can see progressive disease for 2 years and unacceptable toxicity. So they have a Bohr score for each tumor cohort where we can see the response and non-response for each cohort, NIN, GIN, and UGB with the different patient counts. And what we got was they were able to take the stool collection, DNA extraction, sequencing, QC. So what they did is extracted the DNA, sequenced it, and quality control, QC, and read mapping, as well as assembly, non-coding metagenomic assembly genomes, and addition of non-coding NC, GT, DB genomes, which are the metagenomics of this cohort. So this is pre-treatment fecal samples that were collected in 107 trial participants, and they were subjected to DNA extraction, shotgun metagenomic sequencing, and analysis using genome resolved metagenomics. So in this entire pipeline, we're able to see the the metagenomics that were evaluated for the microbiome, as well as the quality control from de novo assembly for near complete MAGs or metagenomic assembly genomes using precise read mapping. And the bar plots, we can get the RECIST score, which is the best overall response with the histology cohort for microbiome evaluatable patients, and the percentage of patients with the objective response, which are partial response or a complete response, are indicated. So these essentially are the clinical and gut microbiome compositional differences between responders and non-responders. They followed them up for 2 years. And we can see the progression-free survival by best overall response group, as well as the overall survival for the best overall BOR group. And we can see the Shannon diversity index, alpha diversity, as well as the beta diversity, and the Atchison distance. So we have a Permanova and an R2 score, or we can see the CR, PR, SD, PD, and CPD in the beta diversity. And in the alpha diversity, we see a tau score as well as a p-value. So the highest Shannon diversity index for the complete responses is at 4. For the partial response, it's at 3.5. For the SD and CPD as well as PDs, it's lower or around 3. These ellipses depict 0.8 for each group multivariate T distribution. So we have the permutations are indicated by the per-MANOVA p-value R squared. Next, we have identification and metabolic potential profiling of the top 22 responses, predictive strains. So the 15 negative strains and the 7 positive strains by strain importance, we can see the density here in this plot. And the impact and prevalence here in this plot on B. And then in C, we can see the prevalence, the impact, and family of the microorganism. We see Lactospirae, Oscillospirae, and Ruminococciae. So in A, we have the kernel density plot, the impact of strains in the strain responder versus partial response classifier with the top 22 strains and the absolute impact for half maximal value. And then the strain impact versus prevalence in the CA209-538 cohort. We can see the strain impact as well as the principal coordinate or the Jacquard similarity of the metabolic pathway that's present or absent for the top 20 strain genomes. So the points are colored by the impact and the size, or the size by prevalence, how prevalent it is, and then the principal coordinate as well. Here we have a phylogenetic tree of the different bacterial strains for a custom reference library, where we can see the 1,391 strains and 6 archaea, and highlighting the top 22 strains. So these are labeled by feature importance for responders versus predictors, or the predictions. A full response or a partial response. You can see at the top Bifidobacterium longum, as well as Clostridium fessum, Avimicrobium case quorum, and F. Praunitzi, Faecalbacterium sp, and Ruthabacterium lactiformans, all in this phylogenetic tree of bacterial strains. So the different colors here, mostly Firmicutes. They dominate the gut microbiome strain response signature. And in yellow, we have Bacteriorhodota. And in purple, we have the Proteobacterium. So 3 different families, 4 different phylums, and we can see the overall impact for each one in this phylogenetic tree. Here in B, we see a prevalence. So F. Praunitzi, R. lactiformans, and A. cicorum, and A. mucinophila. So different bacterial strains. We see the distance and prevalence for each one, and the butyrate acetyl-CoA pathway. So we have the buc abundance in RPM, and the patient R versus P for each response. So the full enzyme name for acetyl-CoA is acetyl-CoA acetyltransferase for the THL here in this heat map, where we can see beta hydroxybutyl-CoA dehydrogenase, which is labeled BHBD, and then CRO or CRO is crotonase. BCD is butyl-CoA dehydrogenase. And the alternative terminal enzyme is butyl-CoA, which is acetate-CoA transferase, BUT, or BUT. And the buc is the butyrate kinase. And then in the box plot, what we see is the but and the buc abundance, and we have acetyl-CoA terminal enzymes. So it's but plus buc, and they're split in patient response, where we have P as progression, 51, where it's 51, and then responders are at, and this is the schematic for the Supervised Machine Learning Framework. Where we see the feature sets, and in the feature sets, we have family, genus, species, and strain, and in clinical features, we have clinical plus each one, so clinical, family, genus, species, and strain, and they're all fed into the Supervised Machine Learning Framework, where we repeat 20 times a 5-fold cross-validation, and the pre-processing includes standard scaling, one-hot encoding, random oversampling, and model training which we use a random forest machine learning training framework, or RF. It's called RF model, and we test it with an ROC AUC model testing. So we use standard scaling and one-hot encoding for the model testing and a random forest for the training. So we get a train and a test together for the supervised machine learning framework, where we input the features, clinical, microbiome, or combined, and the target variables for R response or P progressive. And we get a PFS, progressive free survival 12, that are split into five folds, training folds, one testing fold, which was repeated 20 times per iteration with the AUC score, which is used to select the best hyperparameters. And then what I was able to do is generate these specific sensitivity plots with ROC curves for the strain R versus P classifiers that are retrained using Leave-one-histology-cohort-out, as well as out-of-the-bag subset. So we have a left test out test group and then an out-of-the-bag subset, where you can see these three distinct cohorts. A gynecological cancer, which has a AUC of 0.81, UGB, which is 0.81 as well, which is a urinary gastrobiliary cancer, and then a neuronal neoplasm, which got a 0.64. And we have a mean or average ROC at 0.75, and then a chance, which is able to make the best fit line for the sensitivity ROC curve. So it did pretty well, just a little under the standard. At 0.81 as the top score, we like to see it at 0.85, and then the mean ROC is 0.75. So for this cancer microbiome combined immune checkpoint inhibitor strain resolution, gut microbial signature, it outperformed clinical predictors and was able to cross-validate across tumor histology types. So it did really well for predicting the strain resolution, gut microbial signatures, or GOMS. It was able to outperform clinical predictors, but it just did a little bit under the score. It wasn't able to achieve 8.5. It got 0.81. So we're looking for a 0.85, and we were able to get a 0.81 in two of the cancers, and then in the neurological neuroendocrine neoplasm, it did a lot worse. So the gynecological cancer, as well as the urinary biliary cancer, UGB, he got a 0.81 in the left test out. So that one did better. The out of the bag was lower. Out of the bag was for the gynecological cancer, got 0.73. The neurological or the neuroendocrine neoplasm got a 0.64, and then the U 0.83. So there, the UGB did the best out of the bag, 0.83, which is a little higher than the left test out group. So comparing these two sensitivity tree plots, we're able to see the combined immune checkpoint inhibitor, combined immunotherapy, cancer microbiome, where we were able to model the training and test repeated 100 times with predictions averaged to account for model stochasticity. So what does this tell us? Essentially, we have the fecal gut microbiome shotgun metagenomic sequencing data of these patients, where we have 1,397 quality controlled near complete study specific genomes that can be used as a custom reference database. And in the specific cohort clinical metadata, there's strain abundance data is necessary to replicate our analysis of the baseline fecal samples from unique, richly annotated phase 2 trial charts of patients that have diverse rare cancers treated with combination ICB, immune checkpoint blockade, and 106 discovery cohort, and also the ICB targeting programmed cell death protein PD-1 and cytotoxic T lymphocyte protein 4, CTLA4. It could induce a remarkable yet unpredictable response across a myriad of cancers. So here, we didn't really focus on the cancer type. We focused more on the regimen, on the CICB. and we're able to see the relationship between cancer patients and the gut microbiome composition and clinical response to ICB, yet defining the microbiome-based biomarkers that generalize across cohorts, the GYN, NEN, and UGB, remains a challenge. However, using the ML predictions, we have an improved IC response in PFS relative to models, building using species that rank the quantifications or comprehensive pretreatment clinical factors. And the gut microbiome diagnostics or therapeutics are tailored toward the ICB treatment regimen and is more concordant to the cancer type. So the objective was to get the response versus progression, and that was assessed with the RECIST BOR of the CR and PR, complete and a partial response, versus the PD or CPD, which is the progressive disease primary endpoint. And then we have the BOR of stable disease, SD, and 29 patients were stable at stable disease. And given its ambiguity in pan-cancer cohort, which may represent the disease control or simply indolent cancer behavior. I wanted to include the meta-analysis with the world map, and that included these circles, pie charts, which depict the coordinates of recruiting sites. So these pie charts depict the proportion of patients with tumor response, progression, or stable disease, and the area depicts the sample size. So we're able to see here the sample size. In the 2022 Simpson cohort, there's 38 patients. Excuse me. There's 38 patients. 84 were able to show response, and 84% showed response, and 16% showed progression. And then in the cohort that I was able to show you, the CA209-538 fecal cohort, there was 106 patients where we saw progression disease in 48% and a response in 24%. So here we can see the longitude and latitude of the different cohorts. And also, so that's in Australia. And then in Europe, we see the 2022 Lee cohort, where we see 40% and 44% response and progression. In South America, we can see the 2017 Frankel with a cohort that we have 48% and 38% response and progression. And then we have 2021 Andrews cohort, where we see 64% response and 26% response, or 26% progression. And then in the United States, we have 2018 Matson, 39 patients with 38% response and 30% progression. And then 2022 McCulloch, we see 56% response and 22% progression. It's harder to see the stable disease in these plots, but they're there as well to fill in the space of the pie charts. And then in B and C, we're able to see these tree plots are sensitivity and specificity plots, where you can see, or I was able to see the combo ICB, immune checkpoint blockade, as well as the PD-1 alone sensitivity and specificity. And in this AUC curve, AUC plot, AUC heat map, we can see both the combination, the combo, as well as the PD-1. So the CICB and PD-1 for each test set or each cohort training sets, which one had the highest. So we can see 0.67, 0.78, 0.75 for the fecal cohort CA209-538, and that distributed across each test set and each of the continents. And only a specific subset of patients, 37 with stool collected within 15 days of the start of ICB therapy, was included for the metadata analysis. And then we're able to see the panels that are in the ICB regimen. So anti-PD-1 monotherapy as well as CICB, the combination immune checkpoint inhibitors. So the CICB response could be relative response that's robust across diverse cancers for routine clinically available data. So all of these are comparable with shotgun metagenomic cohorts that analyze over the course of days of the ICB commencement, and fecal samples showed microbial signatures that may be ICB regimen specific. And we can see the different response, SD, stable disease, as well as progression. And in the discovery cohort, we have 7 studies that recruited participants from 11 cities across 5 countries. The countries that were included are United States, United Kingdom, Netherlands, Spain, and Australia. And they represent 470 total patients after they excluded with the best overall response of the stable disease. And the QC reads were mapped to the same reference library that estimated abundance for the same 1,397 strains. So this was able to describe not only the combination immune checkpoint blockade, but also the distinct baseline for the tumor immune microenvironment signatures and the external performance of the CA209-538 strain response signature that's short of what is required for clinical use. And its performance was remarkably better in the combination ICB, where we see the AUC of 0.67, 0.4, 0.78, and 0.75 than the monotherapy anti-PD-1 AUC of 0.46, 44, and 58 and 54 in melanoma cohorts from other industrialized countries. So the strain response signatures were consistent with this trained and on external cohorts, and they were superior when testing the mordant rather than the discordant regimen cohorts. This gives us a strong case for distinct microbial consortium that underpinned their response or non-response to each regimen. So we should really be using this as a standard therapy along immune checkpoint blockades. And given that we know the CICB has a distinct mechanism of action compared to that of the anti-PD-1 monotherapy, it makes the regimen biologically plausible. And by adding the anti-CTLA4, it has a profound effect on the gut barrier permeability that could potentially change the influence of the gut microbiome on the ICB response. Yet, the limitations of the study was that its poorly generalizability of the cohort strain versus RvsP signature, and the anti-PD-1 cohorts are still intriguing. It's something to look into, but given the similarities of the positive strains and those species that are generally previously associated with the response, when using the anti-PD-1 monotherapy in patients with melanoma, Faecalibacterium was linked to the efficacy. And then in hepatobiliary cancers, we have B. long that was linked to the efficacy in melanoma as well as lung cancer or non-small cell lung cancer. And the distinction of these may lie in the negative taxa. So there's many of the top negative strains with our signature that are members of Lactospiraceae family that are broadly associated with anti-PD-1 response in melanoma cohorts in the McCulloch cohort, where we can see the specific strain, the Lactospiraceae in the Lactospiraceae family. All right, so that's it for this combination cancer microbiome immune checkpoint blockade study. I hope you enjoyed the talk here, and feel free to reach out to me for any questions. Thank you for your time.